Topic for the present video is dissolution specification setting, methodology and criteria basics. I have made many of the videos on dissolution testing, dissolution specification, what are the criteria and methodology, and these are available onto the channel. But still, as the dissolution is a vast topic, I thought to make the video so that in the single video the viewers can get idea about the dissolution specification setting and the criteria. So this is a short video, and in this video you will get complete information about the dissolution topic. So as we know that dissolution is a performance test and it is a quality control test also. This test is must to be performed onto the solid oral formulations and also some other formulations where the drug release is important or dissolution of the drug is important for the pharmacological effect. So dissolution is a specific for the product. It depends on to the formulation design, the excipients and also onto the API BCS class. Dissolution is a quality control test, and this test is the only test in the quality control for oral solid formulation, or you can say for some of the oral liquid formulations, which can detect the issues with bioavailability and the bioequivalence. So safety and efficacy is directly related to the dissolution. Dissolution is required to identify the potential issues or problems or would be problems, if any, with the in vivo bioavailability and bioequivalence through the in vitro testing. So dissolution is a in vitro testing and it can identify or you can relate the findings of dissolution testing with the bioavailability and bioequivalence problems. Dissolution testing is done to ensure the batch to batch uniformity, batch to batch quality and the consistency. Dissolution testing is a quality control test which is required to be complying to the specifications for releasing the batch in the market. Coming to the deriving dissolution specifications for, so if you are working onto the new product or you are working onto the novel formulation or innovative formulation, so considering the clinical batches data, pivotal bioequivalence data, pivotal bioavailability data of the batches, you can derive the new product dissolution specifications. So whenever you are working onto the innovator formulation or new formulation, or you are working onto the formulation which will be the reference listed drug or reference formulation, that time consider the dissolution data of clinical batches and the batches used in the bioavailability and bioequivalence studies, and based on that derive the specifications. Also consider the dissolution data of batches failed in the clinical studies and the batches which are complying in the clinical studies, so that you can understand what is the difference in the dissolution. If you are working onto the generic product, then always have the data of reference formulation and also have the data of the dissolution data of the batches which undergone the bioequivalence studies. So apply all the related guidelines and then derive to the dissolution specifications. Also, there is a requirement of discriminatory power of the dissolution method. So that is required to be considered whenever you are providing the dissolution specification, whether it is new formulation or generic formulation. Always consider the formulation design and the release requirements or release claims. Always know that what is going to be claimed for the release, whether it is immediate release, whether it is sustained release, extended release, delayed release, or biphasic release. Consider the API solubility and BCS class whenever you are going to derive to the dissolution specifications. Consider the guidelines for dissolution provided by ICH, WHO, FIP guidelines, guidelines in the USP, guidelines in the BP, EP, the monographs, guidelines provided by EMA, guidelines provided by US FDA, and the country specific guidelines. Consider pivotal bioavailability, bioequivalence batch data for deriving the specifications. Also consider the data of scale-up batches, challenge studies data, and exhibit batches and stability dissolution data. Also consider dissolution data of different packs so that you can give the particular dissolution specification. There are different type of dissolution specifications like Q point specification or single point dissolution specification. It may also be 2 point or multiple points, or it might be a dissolution profile. So know your product design, know the requirements of the regulatory agencies, and then provide the dissolution specification. So Q point specification is generally given for the immediate release formulations. Then 2 point dissolution specification is given for formulations which contain low soluble APIs, so that you need to control the dissolution at 2 points. For example, let's say around 50% or 40% dissolution at one time point, and other time point or second time point where you are getting the 80% around dissolution or complete dissolution. So I have made a separate video on 2 point dissolution, so that can visit and get the good idea about 2 point dissolution specification. Then dissolution profile or multiple time points. Like in 2 points dissolution specification, you can also consider the delayed release type of formulations where you have to provide the dissolution specification for acid stage release that is not more than 10%, and in the buffer stage you can consider the Q point like IR formulations. If the product is extended release, prolonged release, or sustained release where the release is modified, there you have to do the dissolution profiling or multiple time points. Also, dissolution profile or multiple dissolution time point study is to be done for demonstrating the dissolution similarity, for getting the bioweaver, and for the SUPAC changes. So these are the types of dissolution specifications. Reference for dissolution method and specifications. So the important thing here is to understand that what type of dissolution method, condition, and parameters are to be used. First of all, need to search the monograph. If monograph is available, that time monograph to be followed. Consider the market for the formulation, whether it is US market, Europe market, domestic or India market, or any specific country. So search the monographs to get the idea about dissolution and dissolution specification requirement. If monograph is not there, then you can get some idea from OGD database, that is Office of Generic Drugs, USFDA database. Also do literature on reference formulation and other similar formulations if available. If you don't get the information and if monograph method is not there, develop a dissolution method in-house and perform all the uh, studies which are required to prove the dissolution condition selection of dissolution condition parameters, and the specification. Always follow the standard dissolution test parameters and compare the reference product and the test product. So this is for the reference for dissolution method and specification. Then formulation specific dissolution method and the specifications. As I have mentioned earlier, like you have to develop the dissolution method based on to the formulation characteristics. Know your formulation, whether it is delayed release or enteric release or immediate release, and based on that perform the dissolution testing. For example, for delayed release or enteric coated products or gastro-resistant formulations, two-stage dissolution should be there and two specifications will be there in the acid stage and in the buffer stage. If the product is combination product, then see whether single or multiple points to be considered for dissolution, whether multiple drug substances are there or the same drug is having different release. Also consider whether the multiple APIs are layered in the formulation. So consider the dissolution specification and parameters based on to the formulation design. Then multiple release. So some formulations will be having the biphasic release like immediate release followed by delayed release, or immediate release followed by extended release, or multiple medias will be there for performing the dissolution testing. If hard gelatin capsules show cross-linking, then two-tier dissolution will be there with and without enzymes in the media. Media change or pH change may be there, or two medias may be used for the dissolution testing of some of the formulations considering the formulation design. Always consider the bioequivalence batch dissolution data for providing or deriving the dissolution specifications. Some guidelines are there and those guidelines are required to be followed. Regulatory authorities are asking, and in the past also they were asking about the dissolution condition selection, parameter selection, and the specification. Also, the discriminatory power is required to be proved for the dissolution method, whether it is a monograph method or whether it is a in-house method. Always remember that the discriminatory power of the dissolution method is required to be proved or demonstrated. Whatever the method you are using, whether it is similar or same as per the monograph method, or whether it is in-house method, or whether it is similar to the monograph method. So this is the information regarding dissolution specification, setting, methodology, and criteria basics. Basic information I have covered in the video. I request you to watch videos which are available onto the dissolution topic on Pharma Learning In Depth channel, so that you will get good idea about the dissolution. Thank you for watching the video, and I request you to please share this video with your friends and colleagues. Thank you.